Hello, dear students. Have you ever left a bunch of fresh leaves inside a plastic bag and noticed how the inside turns misty with droplets? That's not magic, it's science in action. Those droplets come from transpiration. But here's the challenge: transpiration itself is invisible. So, how do we prove it? Well, scientists and botanists design clever experiments to make the invisible visible. Today, we'll explore these demonstrations and uncover how plants reveal their secret water loss to us. Let's start with a simple yet elegant experiment. Take a well-watered potted plant, place it under a glass bell jar, and keep a strip of cobalt chloride paper inside. Now, cobalt chloride paper is special. It's blue when dry, but turns pink in the presence of moisture. After some time, what do we see? The paper turns pink. Why? Because water vapor released from the leaves through transpiration has moistened it. This experiment gives us direct evidence plants do release water into the atmosphere. Here's another demonstration. If we place a leafy plant under a tightly sealed bell jar, making sure the soil surface is sealed with Vaseline to block evaporation, and leave it for a few hours, something amazing happens. Droplets of water start to appear on the inner glass walls of the jar. And where did they come from? From the leaves themselves, transpired water vapor condensed on the cool surface of the glass. Isn't it fascinating? The leaves act like tiny fountains, constantly releasing water vapor into the air around them. Now let's move to a more scientific instrument, Ganong's potometer. A potometer is designed to measure the rate of transpiration indirectly by tracking how much water a plant absorbs. Here's how it works. A freshly cut leafy twig is fixed in the apparatus, which is filled with water. An air bubble is introduced into the capillary tube. As the plant transpires, it pulls in more water through the xylem, and the bubble moves forward. The faster the bubble moves, the greater the rate of transpiration. Isn't it amazing that such a simple bubble can reveal so much about a plant's physiology? But wait, there's something important to remember. The potometer doesn't measure actual water lost by transpiration. Instead, it measures water uptake. Why? Because not all absorbed water is transpired. Some is used in photosynthesis, some stored in cells. Also, if the shoot isn't cut underwater, or if the sealing isn't airtight, results can be faulty. Still, despite these limitations, potometer experiments remain powerful tools to compare transpiration rates under different conditions like light, wind, or humidity. Or, let's make it even clearer with a comparative setup. Imagine two identical plants under bell jars. One has its leaves coated with Vaseline, the other is left uncovered. After a while, you'll notice the plant with Vaseline-coated leaves shows little or no transpiration effect, while the uncovered one clearly does. This proves something vital: stomata are the main pathway of water loss. And by changing conditions like sunlight or airflow, we can show how the environment directly affects the rate of transpiration. So, what did we uncover today? Although transpiration is invisible, experiments make it visible. The cobalt chloride test shows moisture with a color change, the bell jar experiment reveals condensed droplets, and Ganong's potometer helps us measure water uptake linked to transpiration. Each of these experiments proves that leaves are constantly releasing water vapor into the atmosphere. And beyond just proving it, they also help us understand how factors like light, wind, and humidity affect the process. Isn't it wonderful that something as invisible as water vapor from a leaf can be demonstrated so clearly with clever science? I hope this session gave you a new appreciation for how experiments make hidden life processes visible. Biology truly is about uncovering the secrets of life. Until next time, keep observing the living world around you. You'll be amazed at what it reveals.